right, so just to make sure, I'm not sure what Miss Cynthia covered with you guys, but please make sure you have all of your ingredients on hand if you're going to be participating right along with me. And again, I heard Miss Cynthia giving you guys some background information about DNA, but just a few slides quickly. So DNA is deoxyribose nucleic acid. And again, it is the carrier of all of our genetic information, and it's found in our cells. So animals, plants, and bacteria cells, they all have DNA. But bacteria cells are called prokaryotic cells because their genomic DNA is not encapsulated in the organelle called nucleus. But plants and animal cells, they do have a nucleus. So plant cells and animal cells, if you notice, they have something called a nucleus. And the nucleus is the organelle where our DNA is stored. So plants and animal cells are called eukaryotic cells. So you're considered an animal made out of animal cells. So you would be considered a eukaryotic organism. And a good way that I remember that is that you, as in eukaryotic, you have nuclei or nucleus is in your cells. And again, remember, bacteria do not, and they are called prokaryotes. Cool? All right, and where is our DNA found? Again, like I said, it is in the nucleus of our cell. Most of our genomic DNA, or what we call nuclear DNA, again, is found in the nucleus. You do find DNA in other organelles, such as the ribosomes, but we'll get into that a little bit later. And again, like us, animal cells or eukaryotes, we have enormous genomes or tons of genomic information to store in each cell. And if you guys can imagine, there are more than 3 billion, billion with a B, 3 billion base pairs. Remember that guanine, cytosine, adenine, and thymine that Miss Cynthia just reviewed with you? There's more than 3 billion of those base pairs in one cell. And now, if we stretch those 3 billion base pairs all the way out, that is going to be more than 6 feet of DNA inside of your cell. And 6 feet is about 2 meters, and that's just in one cell. So cells are very tiny and microscopic. You need a microscope to see them, but just imagine they have so much DNA packed in there that if we stretched it out, it would be 6 feet long or 2 meters. All right, now how are we going to get the DNA out of the cell, right? So today we're going to be using a lysis buffer. And lysis buffers are used to break open not only the cells, but then break open the nucleus as well. And most lysis buffers contain detergents and salts, such as sodium chloride, and they help to keep or balance the osmolarity or water molecule distribution of the lysates. And if you could see in the picture, we have an intact cell in the top left. And then when we add our lysis buffer, we're first going to rupture the cell membrane or the cell wall that's surrounding the cell. So you now see how there's a hole in the cell membrane. But now we also have to break open the nucleus to get the DNA out. So then the detergent starts working on lysing the nuclear membrane. And then your nucleic acids, your DNA, will come out of your nucleus. And lysis buffers can be used on plants and animal cells as well. All right, so let's get into our activity. I know that we are waiting, have been waiting and anticipating. Again, thank you for being so patient. And now give me another second to start our video. Video. Okay. So does everybody have their reagents close? Cool. As we talked about previously, we're going to need to break open the cells of the strawberry in order to pull the DNA out or to extract the DNA. In the lab, we use lysis buffers to lyse cells open, and those could be bacterial cells, plant cells, and or mammalian cells as well. So step one in isolating your strawberry DNA is going to be to mix or make your lysis buffer. So to make your lysis buffer, you're first going to need a container to put it in. Then we're going to need some dish detergent. We're going to need some water to dilute our dish detergent. And we're also going to need salt. In a second, I'll explain why the salt is so important. And then here I have a graduated cylinder so I could measure out the proper ratios of our ingredients. Then I'm going to add them to my container and mix together. And again, this is the lysis buffer that we're going to use to lyse or break open our strawberry cells so we can isolate or take out the DNA. All right, let's make our lysis buffer. So your lysis buffer is mainly dish detergent or soap and water. And we're gonna mix our soap and water in a 9-to-1 ratio, meaning that you have 9 parts of water and 1 part of dish detergent. So for today, I'm going to do 1 milliliter of dish detergent to 9 milliliters of water. And then we're gonna add in a pinch of salt. And a pinch of salt is anywhere from 1/4 of a teaspoon to 1/2 of a teaspoon. And just for simplicity, I'm gonna make a total of 20 milliliters today. So I'm going to be adding 2 milliliters of soap and 18 milliliters of water. Okay, just to clarify really quick, you guys, as long as you keep your ratio 9-to-1, you should be fine. And 1 milliliter is about 1 teaspoon. So if you put 2 teaspoons of soap, make sure you put 18 teaspoons of water. Cool? Or 1 teaspoon of soap and 9 teaspoons of water. And then your pinch of salt, which is 1/4 of a teaspoon. But when I say milliliter, your kitchen equivalent to a milliliter is 1 teaspoon. So right now I'm doing 2 mils or 2 teaspoons of soap and 18 milliliters or 18 teaspoons of water. Let's start with the water. So right now, measure out your water. All right, that's about 18. Let me get a paper towel to clean up my mess.
Can you say that ratio again?
So it's 9-to-1. So if you use 1 teaspoon of soap, you should use 9 teaspoons of water. Everybody got their soap and water mixed together? All right, so you can now see my final volume is the 20 mils that I was heading for. And now for our pinch of salt. Again, a pinch of salt is about 1/4 to 1/2 of a teaspoon. All right, and now we're gonna get our secondary container. swirl to mix in our soap so it just doesn't stay in the bottom. All right, and now we can get it all in here. And you guys can feel free to mix your lysis buffer in the container that you're going to store it in and use as well. And now that this is combined, again, I'm just going to keep swirling to mix. You really don't want to shake because this is soap, and soap forms bubbles whenever you shake it up. So, okay, we're swirled to mix. All right, so we always like to label our bottles in lab, so I'm just going to take a second and label this lysis buffer. With my initials, so they know I made it. Be able to break.
Sorry for the technical difficulties. Clearly this isn't my edited video that I made for you guys, so I'm gonna go ahead and skip through some of the parts.
A second and label this.
All right, does everybody have their lysis buffer made? So we have soap, water in a 9-to-1 ratio with a sprinkle of salt. Cool, thank you. Now everybody grab your strawberry and set it in front of you. Again, your alcohol should still be in the freezer, but right now you're gonna need your strawberry, a Ziploc bag, and your lysis buffer.
So first things first, we have a strawberry packed full of DNA, right? And we want to extract that DNA or pull that DNA out of the strawberry cells. So we learned a little bit about cells and their chemistry and how they're made up. So both animal and plant cells have a cell membrane, but plant cells also have a cell wall on the outside of that cell membrane that makes them nice and rigid and tough. So the reason why we had to make our lysis buffer was to be able to break open that cell wall, break open that cell membrane, and also break open the nucleus where the DNA of the cell is stored. So now we are going to add our strawberry into a plastic baggie and add some of our lysis buffer. Then we're going to give our strawberry a gentle squeeze for about 3 to 5 minutes, just so all of our strawberry is mashed up. Let's get into it. And I'm using 1 strawberry. 1 strawberry is enough, but you could feel free to use 2. But remember not to use too many, as these are food and we don't want to waste too much. So I have my strawberry in my bag. And now we're gonna add a little bit of your lysis buffer. I made 20 mils, so I'm gonna add about 5 to 10 mils, or about half of what I created.
So you guys, 10 mils is enough if you want to add 10 mils or 10 teaspoons of your strawberry lysis buffer. That should be good.
All right, now what's key to mashing up your strawberry really well is removing most of the air out of your Ziploc bag before you seal it up tight. And that's gonna help you just to mash your strawberry really well. So my strawberry was actually a bit soft already because I did get it from the freezer. So feel free to use frozen or fresh strawberries to do this. But as you can see, oh, it smells so good. I can smell it already. But I'm just squishing the strawberry into the lysis buffer here. See?
So you guys, if your strawberry wasn't previously frozen, go ahead and squeeze really hard, or put the baggie down on the table and squish the strawberry kind of with the palm of your hand. Just make sure it's all squished up.
Set it down, squish this way. So right
Good
now,
job,
the
Becca.
detergent in our lysis buffer is breaking up all of the strawberry cells. Again, the cell wall and the cell membrane. And it also has to break open the nucleus because that is where the DNA in our cells are stored.
So keep mushing with your hands. Keep going, keep going. We want to break open as many cells as we can.
Right.
The more cells that we break open, the more DNA that will leave the cells and we'll be able to isolate and collect at the end.
So I think we're pretty good here.
All right, people still squishing. I see a few people still working, so I'm gonna give you a few seconds to squish your strawberry. Make sure it's very well smashed up, just like in my video. All right. We're good to go. All right, keep squishing, keep squishing. Good job.
So just keep smashing until your strawberry lysis buffer solution now looks like this. So now that we have our strawberry nice and mashed up in our salty, soapy lysis buffer, we are now going to have to separate those solid strawberry seeds in pieces from the lyse strawberry juice. So to do that, you're going to want to grab a coffee filter and a funnel.
If you don't have a funnel, you can use a strainer. That's fine as well. You just want something to hold your coffee filter.
Also a container to catch the juice that is strained out. So once you have your container, you can place your funnel inside and then grab your coffee filter and then place it down inside your funnel. And the coffee filter, again, is going to catch all the solid parts that weren't dissolved from your squeezing of your strawberry and the salty, soapy lysis buffer. Go ahead and open your Ziploc bag and try to get everything kind of like to one corner, one side. And then you go ahead and pour it on out. You could go ahead and squeeze the tail end of your Ziploc bag just to make sure you get everything out.
Okay.
And now, if you guys have opened your Ziploc bags, you can definitely smell how good this smells. Nothing like strawberries and soap, huh?
Smells great, guys, huh?
All right. And some of my strawberry juice is already beginning to drip out, but what you guys can do to help this move forward a bit is to go ahead and close up the top of your coffee filter and just like the Ziploc bag, start squeezing down from the top. And give your coffee filter a gentle squeeze, very gently, because if you squeeze too hard, your coffee filter will break and then all of your solid pieces will end up back in your strawberry juice and you'll have to start all over. So just give your strawberry a gentle squeeze in the coffee filter just to get the majority of that juice out. And again, the juice we're squeezing out is going to be the insides of the strawberry cells, the salt that we added to the lysis buffer, the soap that we added to the lysis buffer, and some water. Let's just take our time and squeeze. And after a while, gravity would do this, but that would honestly take a couple hours. Just go ahead and keep squeezing very gently. Okay, I think I'm just about done. All right, so now you can see that in your coffee filter should be all your strawberry solid pieces, especially your seeds. But down in your container, you should have a lovely strawberry colored looking juice. And again, this juice here contains not only our DNA, but all of the contents of the strawberry. So strawberries also have DNA, RNA, proteins, lipids, and all other molecules that are found in the cell. So moving forward, we're now going to want to just separate out the DNA from the rest of the strawberry cell's contents. So again, moving forward, we're just going to isolate or extract DNA, and we're going to pull that away from the strawberry cell's lipids and its proteins and other molecules. So now that we have lysed our strawberry and we have the strawberry cell contents, again, inside of our lysis buffer, now in this flask, what we actually have are strawberry proteins, strawberry lipids, strawberry carbohydrates, strawberry DNA. We also have detergent or soap, and we have salt. So now what's going to be useful for us is the salt. So again, we're only here to isolate strawberry DNA, but I said there were a whole lot of other molecules hanging out with the DNA in here. So how are we going to separate just the DNA from the rest of the cell contents? So because this DNA is now in a salty solution, we can use super cooled alcohol to extract the DNA out of this solution. All right, everybody run to the freezer and grab your alcohol really quick. This is the time we're gonna need it. Good job, good job. And this is because that salt will help the DNA to clump together and actually move into the alcohol and out of the salty lysis buffer. So how we're going to do that? We're going to add the same volume as in this container, add the same volume of that cooled alcohol that
Now I'm talking.
So I have about 10 mils in here. So I'm going to pour another 10 mils. So the same amount of lysis buffer that you guys use, you're going to want to match that amount in your alcohol. So if you use 10 teaspoons of lysis buffer, you're going to want to use 10 teaspoons or 10 mils of cold alcohol. All right. Now, in order to do this, we're going to have to pour very slowly. So because the bottom of my flask is very wide, I'm going to tilt mine to the side so it collects right here and it's a smaller surface area. And then I'm going to slowly pour in my alcohol so that it forms 2 layers. So you want to pour very slowly. So remember how Miss Cynthia poured slow last week for the density tower? So remember how you guys poured really slow last week? So just pour your alcohol really slowly on the side of your container so that it layers on top of your lysis or strawberry lysis buffer. Mix, but instead they form two layers. Do you see the two layers forming? All right. So now that our alcohol is in there, we have those two layers forming. Now, what we should start seeing after about a minute is some cloudy kind of residue raising up into that clear top portion, which is going to be our alcohol. And guess what that cloudy stuff is? So we should see cloudy stuff leaving the pink juice layer on the bottom and entering the clear layer on the top. And this might take 1 to 2 minutes. I'm gonna go ahead and gently straighten up my container as to not disturb those two layers and set it down.
So again, guys, this takes a few minutes to happen. You might see something as soon as you add your alcohol on top of your strawberry lysate, but give it about 1 to 3 minutes, and then you should see a white stringy substance rising into the alcohol layer of your experiment. I'll show you a close-up in a sec.
And you guys should be able to see something cloudy or stringy rising out of the strawberry juice and into the clear layer. So we're gonna let this sit for another minute or two and then make some observations. So again, we're just letting our very cold alcohol sit on top of our strawberry juice lysis buffer solution. And remember when I said you guys should see those opaque strings popping up? Well, now you can actually see them in the clear layer. So the alcohol is a clear layer on top here. And again, our pink strawberry juice lysis buffer is on the bottom. And now you see those kind of white stringy substances.
And it does tend to stick to the side of your glass container.
So what do you guys think that is?
Does everybody see that white kind of stringy substance in their alcohol? Thumbs up if you've seen it yet. You do? Good job, guys. Good job, good job, good job.
And again, this should be happening after about 1 to 2 minutes. You can let it go as long as 5 minutes. So again, what we're looking for is now this white fluffy stuff, which has formed after about 3 minutes in the alcohol or clear layer. And again, I will ask, what do you think that white sticky stuff is? Let's move forward and go ahead and grab it out.
Now this is when you need your wooden skewer or your toothpick or your tweezers.
So now that our DNA has been isolated from the strawberry lysis buffer and the strawberry juice, we could now take a wooden skewer and we could take the tip, and what you're gonna wanna do, because DNA is very sticky, is just swirl it around and try to collect all of that white stringy residue that is in the alcohol layer of our DNA extraction. And look at this. So this is all of the DNA that you can find in just one strawberry. That strawberry, that small strawberry, oops, I got it on the side. That small strawberry that I started with. And I'm just gonna set it here in a Petri dish, if I can get it off the metal.
It really didn't work out for me.
Skewer. I guess I can't.
So DNA is so sticky that it would not come off of my wooden skewer.
This is our strawberry DNA. So you guys see that sticky white substance that's stuck on the end of our skewer here? So that is actually the strawberry's DNA. So we successfully lysed the cell open, lysed the nucleus of the cell open, and extracted the DNA using salt and alcohol.
So the next minute or so, I'm going to show you guys how I extended the experiment.
So pushing the experiment out even further, about another 15 minutes, what I did was totally mix the two layers.
You guys don't have to do this now.
Then let it sit and re-separate for another 10 minutes. Then I put my wooden skewer back into my flask, and look how much DNA I was able to isolate. And again, this is just using one strawberry. So one strawberry produced all of this DNA.
Again, guys, too, with this final portion of the video, it took me another 20 minutes to do this. So you guys can go ahead and do it after the Zoom class if you want to continue. But right now, I'm just showing you the total amount of strawberry DNA that you can get out of one strawberry.
Oh, huh? See if I could catch it. It wasn't as sticky as the first batch I pulled out of here. But you guys get the picture, right? This is our strawberry DNA.
So hopefully with all the technical difficulties, everybody was still able to, oh, beautiful, Becca's. Everybody was still able, hopefully everybody was still, oh, great, go River. Everybody was still able to isolate their DNA. Good job. Is that Easton? Long time no see, pal.
Hi, I'm the one that dissected the frog at the opening of that place.
Yes, sir. I remember you. You doing good?
Yes.
Hi. That's good. All right.
I
So
got a whole lot of DNA.
what? You just used one strawberry?
Yes.
Yeah, and that is a whole lot. A whole lot. So again, you guys, thank you for participating, but I do want to say thank you, more so for being patient with all of Dr. Candace's technical difficulties today. So you guys can continue to try to isolate DNA from different fruits in the house. So one of the participants had already asked if we could do this with different fruits. So yeah, you could go ahead and try grabbing a banana or maybe an orange or a blueberry and see if you could use the same protocol and the same lysis buffer to isolate DNA from other fruits. So Miss Carrie Ann just said in the lab, she has successfully got DNA from bananas, plums, and even spinach. So don't use all your vegetables for your experiments. Eat most of your vegetables, but grab a few leaves of spinach and see if you guys can isolate DNA out of vegetables as well. And if you guys want to, you could always take a picture of the DNA that you isolated and share it with us on Facebook, or you can email it directly to me. And I would love to see what you guys are experimenting with at home. So since everybody's good to go, I am now open for any final questions. I know we're a little over time, but I'll open up the message box for any final questions, or you can feel free to unmute yourself to ask a question, and then we will get into the final poll. Yes, Mr. Easton?
Can we look at that under a microscope?
Yes, you could absolutely look at your DNA under a compound microscope. You'll probably only see the thin strands, excuse me, of DNA, but you would actually need a electron scanning microscope to see the actual structure of DNA. And those microscopes are really big and really expensive. So if you know somebody down at Pitt or CMU, give them a call. But yeah, if you have a microscope at home, take a look and tell me what you see. Shoot me an email.
Okay.
Thank you.
Thank you.
Thank you.
I love the class.
Oh, thanks, Ethan. Anybody else? All right, so you guys know we like to end all of our sessions with another assessment, and usually the questions are the same that they were in the beginning, but this is just a way that I can see that my knowledge is reaching into your little brains, all right? So I'm gonna go ahead and launch our final poll. So there's 7 questions, so you could take about 7 minutes or so to complete it. All right, the poll has been launched. And I'll also give you the answers to the poll questions after about 5 more minutes. Again, if you have any more questions, feel free to use the chat box.
And click.
All right, just waiting for one more person to go ahead and finish up. Okie dokie. All right, let's start at the top and go over these questions. Good job, you guys. Everybody did really, really, really well, and hopefully you learned a little bit more about DNA. The last time I talked to you guys, you learned about the structure of DNA and the backbone and the different base pairs. But today we learned more so where DNA comes from and its actual purpose. So the first question is, what is the purpose of DNA? And the correct answer for that is to carry genetic information. So each one of our cells has the genetic information to create our whole body. So each one of those strawberry cells had all the genetic information to create or make and form that full strawberry. So the purpose of DNA is to carry genetic information. All right, question 2. Where is genomic DNA located in a eukaryotic cell? So remember, we are eukaryotes because we are animals and we have animal cells, and animal cells hold their DNA in the nucleus. So eukaryotic cells have nucleus or nuclei, and that is where the DNA is stored. All right, number 3, can you see DNA with your naked eye? Kind of a tricky question. But we're going to go with yes. If enough is gathered together, we can actually see DNA. And that's what you guys did today. So just looking at that strawberry, when all the cells' DNA were separate, we could not see the strawberry DNA. Or even in the lysis buffer, after we lysed our strawberry cells, we couldn't see DNA. But after we did some experimentation and drew all of those DNA molecules together, we were then able to visualize our DNA or see our DNA as that white kind of fluffy, snotty substance. All right, plants and animals are eukaryotic. Very good, Zoe. So plants and animals are eukaryotic, and that means they have a nucleus. And bacteria are prokaryotic because they do not have a nucleus. Number 5, cells can be broken open using a jackhammer. What was I thinking? But yes, the correct answer is lysis buffer. Cells are broken open using a lysis buffer. And your lysis buffers usually contains detergents, salts, and water. So remember, the first step of our experiment was making that lysis buffer, and all we added was detergent, which is going to disrupt the cell membrane, water to dilute that detergent, and a little bit of salt, which is going to help our DNA molecules come together. All right, and the final question, number 7, inside of a human cell, which is your cell, the DNA can stretch out 2 meters. So 2 meters is about 6 feet. So 2 millimeters is the space between my nails. 2 centimeters is about this big, but 2 meters is about 6 feet long. So you have tons of DNA packed into your nucleus of your cell. All right, thank you everybody for participating. I had fun today despite the technical difficulties, and I will be back next week to see you guys. We can get into a little bit of, I believe, a little bit of earth science next week. But I'll hang out here for about 5 more minutes in case you guys have any questions you want to direct to me personally, or you could share with the whole group, or you can go ahead and unmute yourself and ask me any more questions. I'll hang on for about 5 more minutes. If not, see you next week. Thank you guys for coming. It was fun. No questions? See you, Preston.
I'm doing raspberry DNA right now.
What did you put in your bag? Oh, raspberries? Okay, yeah. Preston, yeah. Let me know how that goes. That was a good idea. Let me know how that goes. So you could email me or you could post it on Facebook or anything like that. But good idea.
Okay.